Hello, everybody. It's time for Q&A with A&V. Welcome back. I am Vincent Racaniello, and joining me tonight on piano is Amy Rosenfeld.
Yeah, I used to play 100 years ago.
You did play piano?
Yeah, 100 years ago.
I didn't know that.
I lacked attention. I lacked focus and attention to detail. Then I played tennis. Lacked a focus and attention to detail there too. What can I say?
So I recorded my daughter playing piano, and it's the background since some of you seem to like it. And there it is. She's actually covering a Fiona Apple song.
Yeah, not my favorite artist.
Do you don't like Fiona Apple?
Not really, but that's okay.
Well, she got the sheet. I have to say, she got the sheet music, and is learning how to play it.
It's good. Everybody should learn how to do something.
Okay, folks, do you want an update on the?
So yeah, you tell them your big news.
So what's the big news? Amy and I selected the carpet and the wall paint for the incubator. So here's the state of the incubator.
As of yesterday.
So this is going to be the studio, this space in front of you. There are windows on the right, and there's a wall in behind, and this is going to be insulated and extended. That'll be the door. And most of these wires are going. We don't need so much internet stuff. So that's that. And let me see what else I have. So this is the other part of the office. The main entrance is here on the left. This is the electrical closet. Here's the kitchen. And there's an office over here. We're going to put a conference table here, right, Amy?
Yes.
Somewhere in here. And then there's a little space maybe for a workstation. Unfortunately, this column can't move, so it's kind of in the middle of everything. Let's see what else we have. There's Amy by the windows. And this is the studio part. And it's a little funny. It's got this bump out here. It's just really bad. I thought he was going to take that away. Shouldn't we tell him to take this away?
I don't think he can take it away. I think it's the wall to the office that's next door.
Yeah,
but you could double check.
There's a wall behind it, and it's just kind of bumped out.
Oh, yeah, maybe. All right. We can double check. I forgot. And
one more, I think. Well, I'll have to move it up before we can see it, huh? Yeah, that's just the view. This is the old entrance going to be blocked off. So there's the studio. No water cooler here. That's going. All right, so that's the incubator, and I'm trying to figure out the internet for it because it's too slow. I want gigabit, and they don't have it, so I have to work on that.
You have to call.
Who are you going to call when you have ghosts?
Ghostbusters, of course.
Amy, before you go today, tell us what you're going to do. Last week, you took your cells out of trypsin. What cells, where were they before? The purpose of trypsin. Next stop, possible pitfalls. Like a grant.
Yeah, I don't like this man anymore.
What are you doing tonight? Just tell them what you're doing tonight.
What I'm doing tonight is I'm working on the grant that we're sending off on Tuesday by 5 p.m., which is a very large grant. And it's like my baby, right? I've been cultivating it since February.
Yes, there's all Amy's ideas. She's been doing the preliminary data, and we are writing it together with Ian Lipkin and
Nishay.
And Nishay, who's a research scientist in his lab.
It's quite the undertaking. And, I don't want to forget to thank Kyle for the lovely bottle of Barolo. I'm looking forward to relaxing and drinking it one evening after the grant gets deposited. So, yeah, So
today you didn't do experiments as a result, right?
Well, should we tell the mouse house story?
Sure.
Okay, so I went to go to the mouse house that houses our infected mice, and it's an airlock system, and you go through multiple doors, like 4 or 5, right? Vincent?
Mm-hmm. Yes. 4 or 5 doors. Yeah.
Jeez.
Come out of the elevator. You go through one door. You
Then you
go
go through
through
another door to
another
the
door,
autoclave room.
another
Then you go
door.
through a key pass door. Then you
Yeah.
put in your passcode to the suite with the mice. Then you go. So now you're in the room with the gowns, and you gown up. And then you go through another door to go to the sink room, which you wash up from when you come out. And so I gowned up, and I pushed the button to release the airlock so I could go into the sink room, and the door's broken. Doesn't open.
You couldn't get in.
Couldn't get in. Stood there for like 10 minutes pushing the button. Okay. Then I go upstairs and I say to the lady, I think the door's broken. Doesn't let you in. So she says to me, no, it's probably just that the lady had door 4 open and you can only see door 2. And I'm like, well, that doesn't help me.
Did you get in?
Yeah, but it was like 25 minutes later.
I'm
This
sorry.
takes like forever.
Mike writes, have you done a TWIM on Nature's Find Company and their discovery of a fungus strain that could supply the world with protein? That's actually an interesting TWIM topic. I'll look that up. Haven't done it, but it's a good idea. Thank you very much. I'd like to hear your opinion on why mRNA 12273.351 generates less neutralizing antibodies to other variants than 1273.221. I don't know. These are
mRNAs.
Well, they're variant mRNAs, right? 351 and 221. You know, I learned yesterday from Fouchier that everybody assays neutralization differently. There's no standardization, as there is for in flu. It's a neutralization. It's a hemagglutinin inhibition assay. Everybody uses it. He said, for COVID, nobody's standardized. Everybody uses different things. So who knows what
Theodora
it means?
told you that. Theodora and
she
Paul,
did?
yeah, they discussed it. It's a big problem. And we're finding that it's a problem for our viruses. That reality is that everybody does it differently, such that we think that we received sugar water, and now I have to do 4 times the amount of work to prove that it's sugar water. You know what I'm talking about.
Yeah. I got it. Has the percentage of long haulers increased, decreased with the various variants?
I believe it stayed the same. They haven't really reported that there's an increase in long haulers. And they haven't really said that we know that there's no difference really in the ratio of symptomatic to severe disease because you don't see hospital cases increasing or deaths increasing.
All right.
But the other thing is that the people who are getting it's not really a fair comparison, right? Because the people who are getting infected with the variants are the younger people who had lower risks getting those severe things to go to the hospital and die compared to the older people.
Yeah. All right, Amy, what concerns you about the future of COVID?
What it's done to science.
Could you elaborate? I know what you're saying, but our people here would like to know.
That the profession that I fell in love with leaves something to be desired.
What are you saying?
Has been tainted.
There's been a lot of bad science during COVID.
You think?
Need a T-shirt. You think?
Can I have a T-shirt with the eyebrows raised too? Because you got to do the eyebrows. You think?
Yeah, the eyebrows. Yeah.
Yeah.
All right. So you're concerned about its effect on science. Yeah, I agree.
Well, I think that is the lasting effect. I mean, I'm not really concerned about that. I mean, it'll become an endemic virus, and we'll vaccinate people just like we vaccinate kids against polio and DPT and MMR and stuff, and it'll be controllable. But going back to the way science should be performed and scientific discussion without polarization and critical thinking and stuff, you can't really go back to that. And so that's my greatest concern, is that now you can research any artifact that you want with no repercussions and no consequences and stuff. And that's going to be applied to everything now.
All right, so John said that most of us know what you're talking about. Good answer. All right, so you didn't have to explain. Okay, good. I'm glad you, after coming here for so many months, yeah. Well, my concern is getting most of the world vaccinated. Many countries have so little vaccine so far, and they're not going to do it on their own. It's up to us. We need to help
but there
them.
there was an article that said even the poorest country will be pretty much vaccinated by the middle of 2022.
I hope so. I hope you're right. That's great.
And so that's like, yes, we hogged it all up, but there's also a dearth of manufacturing plants, right? Like the entire country of Canada doesn't really have a manufacturing plant, and now they've decided that they're going to invest and create one. Right? So that's like an immediate short-term COVID problem, but that's not really like long implications of this pandemic.
Well, I hope you're right, but it does bother me. All right, Richard wants to know, we get zoonotic spillovers from animals. Is there such a thing as a veginotic spillover where we get infected from plants? I don't know of any.
For viruses?
Yes, for viruses, yeah.
No, but you can imagine salmonella from whatchamacallit, lettuce and stuff, cucumbers.
Sure, well, we can get bacteria from contaminated. But plant viruses, no, I think they're too...
Well, they're totally different because plant cells are not autonomous.
Yeah, and also they don't bind to receptors. They just get put into plant cells by, insects or nematodes or farmers breaking the leaves and so forth. So they wouldn't be able to get into an animal cell. So I think not. Although you do pass through a lot of insect and plant viruses every day in what you eat. And you may say, insect? I say, yeah, the insects that crawl on the plants in the fields, their viruses go there too. Amy and Vincent, isn't it a good idea for vaccinated people to still use masks to prevent potential long COVID and to protect the unvaccinated and those in whom the vaccines don't work? What do you think, Amy?
I still wear a mask when I go out. I think at some point you won't have to, like there'll be community immunity. But at this point, we haven't reached it. And we probably won't reach it until we make it so that, 2 and above get vaccinated? Is that what the trials are looking at now?
Yep.
Yeah.
Yeah, I think we can get to the point where most people will be protected, but we're not at the level of immunity yet that we need in the U.S. anyway.
Anywhere.
I walk in, when I go around New York, I wear a mask. We have to do it on the subway anyway. New Jersey Transit, in our building. I wear a mask a lot in public places, yep.
Are you taking the train tomorrow?
Yes, I am. I can't drive anymore. I fall asleep at night. I don't want to risk losing all this community. You'd have to take it over on your own, Amy.
It's okay. I have a plan that you've been selling to people as your own, like, my push for you to do fundraising the week right after Thanksgiving, when people have to get rid of all of their money. You know how we did for EV68 for 8668. We did it like the Tuesday after Thanksgiving through the beginning of the next year.
Right. So speaking of fundraising, let me introduce Jeffrey, who Microbe TV has hired to raise money for us.
Your new development resource.
A professional fundraiser. Thank you,
Yeah.
Jeffrey. Looking forward
Good.
to working, and Amy's looking forward to working with you as well.
Well, this is the way it works. I tell you the ideas, then you decide you should email him the ideas, and you say blah, and then he says something, and then you come back and you say blah, and then I refine. That's how this works. That's how this dance works.
How does the blah? The blah means fill in the blank, right?
Exactly.
What do you think of bacteriophages for treating bacterial infections? A lot of people are working on them. We have covered a lot of phage therapy in TWIM. There are some really exciting success stories, but there's still some obstacles to them becoming popular. And, one of them is, they may be developed for surface infections, topical infections, but systemic may be harder because, the phages are cleared rapidly and so forth. So, many people are working on it. I think they will have certain applications. There's already a phage approved which kills listeria, which is a common contaminant of food, and they use it to spray food preparation surfaces. Can you imagine spraying phage on a counter and it's lysing the bacteria? But it's approved. It's Department of Agriculture. Who would do that? Who would do the food? Is that FDA?
FDA.
Okay.
Food and Drug Administration.
Yeah. Let's see here. Let's find another good one. With the statement that T-cell epitopes don't change, how did they do the experiment to demonstrate that?
Read Shane and Seti's paper that came out yesterday.
Well, they come here for answers, Amy. Can we explain it? Okay, you want me to do it? I'll do it. So what you do, Samantai, is you make peptides from all of the viral proteins. You can actually predict what the T-cell epitopes are. That is the sequences recognized by T-cells. You take the peptides, you have a company make them, and Amy knows all about this because this is part of her grant, except for B cells. And then you throw them in a dish with lymphocytes from a vaccinated person, and you see if they react, if the lymphocytes react to the peptides. And that way you can know if there are T-cell epitopes being recognized. And so you do that, and none of the sequences that T-cells recognized are changed in the variants. And that's why you don't get serious disease after vaccination and infection with a variant.
Also, just listen to Ron tomorrow.
Oh, Ron Fouché, TWIV 777, dropping at midnight. You're going to love it. Well, some of you will. And for example, this question, what fitness advantage does the Delta variant have? So Ron thinks it's all... Well, Ron thinks it's all antibody escape.
Can't be all antibody escape because there are other mutations. And recently, Nevin's paper says it's not the spike. So it can't all just be, it has to be investigated. To just say it's antibody escape is a good guess, but it's premature. It needs to be investigated.
Well, we're having a casual conversation on TWIV. He says if this is in flu, the fitness advantage for flu is antigenic drift, and so
but you only see alterations in HA and N.
Yes.
You don't see alterations throughout the entire genome.
Yes.
And here you're seeing alterations throughout the entire genome. So it is hasty to say that it is antigenic drift.
Let me just say, he said, what bothers me is that there are so many other mutations in the genome.
But yet
spike.
You just said he said it's all antigen.
I said he thinks it's antigenic drift, but he says what troubles me is that there are many other changes. But we'll see. We'll see.
It's too elementary to just say it's antigenic drift. It's not.
Yeah, so I played the Nadia music before. I'll play it again. As I said, that's a cover of a song that Amy doesn't like.
I just don't like the performer, but it's fine.
If SARS-CoV-2 is a quasi-species and mutates in almost every cell, Why the most recent virus differs by only 20 nukes from the original strain?
Because all the ones that differed more were lethal.
Yeah, many of them are lethal. Yep. Yeah, the dog is much better. She's now pretty much normal and running around. So they gave her drugs. They gave her prednisone and gabapentin, which, dampens down the inflammation, the neuropathy. So she's good. You sure you didn't copy your glasses from Britt Glounsinger? Does she wear blue glasses? No, I didn't. I didn't have her in mind when I bought these. I just looked at Amazon and I said to Amy, red, orange, or blue? And she said blue. She's my fashion consultant, my business consultant, and my science consultant.
A lot of jobs. That's why I'm old, wrinkled, and disheveled.
Is the UK right to end all restrictions on the July 19th when they have 90% of adults vaccinated and 70% both doses? What do you think, Amy?
Well, 70% is what the prediction for herd immunity is. I mean, I think it's a little hasty. I would like them to be over 70% in case there's some kind of calculation error.
Mm-hmm.
But I mean, yeah, that's the point of vaccinating, is that you can return to life. But the thing is while England may be 70%, the rest of, the neighboring countries and the people who come in and out of England are not 70%. So it would have been better to be a little more cautious, or be more prudent. But I'm not in charge.
Ken wants to know if the first TWIV episode is available online.
Yeah,
it certainly
is.
And Dixon. Here, let me show it to
guest.
you. It was me and Dixon here, and it's stunningly simple in its presentation. There's the TWIV website, right? TWIV 1, West Nile virus, September 24th, 2008. Me and Dixon launch a new science podcast with a description of the emergence and spread of West Nile virus in the U.S. And we had
Dixon's book.
By West Nile Story. We had a couple of links. Look at an episode transcript. Someone transcribed it subsequently. Yeah. And, here's one comment here. Here's where it all started. Thanks. Yeah, so, you can't get all of the episodes on your podcast player. There's some technical reason for that. But they're all on the website. Yeah, for sure. How about talking to Louis Rossman, who has gone through the same build-out? You mean a studio build-out? Sure. I will do that. Yep. What's happening with measures to reduce spread of Delta? Vaccination and vaccination. You could wear masks still. You could distance. Ron Fouché said yesterday, by the way, he said all the bars and restaurants opened in the Netherlands and the cases are going up. So what do you think, Amy?
I think that they're not vaccinated. I know, you probably need a vaccine passport, but, it's on the honor system.
Actually, he said you can't go to the clubs without being vaccinated, but what they do is they PCR test you. So they're not actually looking at disease. They're just looking at PCR positivity. And, of course, people are going to be PCR positive.
Yeah. So how do you do that? You say you're going to go to the club on August 1st and you get tested today for your August 1st reservation?
I don't know. You have to be vaccinated, a good weeks before, first of all. You have to be vaccinated to go.
Yeah, but you just said that they're doing PCR tests.
Yeah, I don't know when
I don't understand.
I don't know. Amy, will the incubator be level 5? BSL 5. Oh, we could put that on the door, BSL 5, under the incubator sign, right? What do you think?
Could be.
Okay, that means Amy is okay
we'll
with
discuss. It. No, it means we'll discuss. That's
Folks,
all it means.
Do you know what we'll discuss means? She doesn't like it. She wants to talk me out of it. Okay, no problem. Thank you, Les, for your contribution. Oh, that's so nice that you're your favorite. We appreciate it. And you can interact with other people here, like-minded people, right? That's really
it's all
great.
good.
Amy, how quickly can monoclonals be produced?
Depends on the virus, because there's a graph in a book that says when you inoculate somebody.
Manufactured, I mean. That's
Manufactured?
Yeah.
Well, it takes a while to clone them out and clone the gene and produce them and demonstrate that they're neutralizing, if that's what you're looking for. It could be a couple of months.
Yeah, could be a few months. But the single chain, the nanobodies, can be produced much faster than
no,
because they can be produced in E. coli, and you don't need to make huge reactors with eukaryotic cells.
What? You still need to find that antibody. You still need to clone the gene.
But production,
you still need to prove production for what? I don't know. Okay, I don't know.
Amy, when you get your antibody, you want to produce it in mass quantity to give to people. It's much faster with an antibody that you can make in E. coli.
Is?
Well, it's a combination. How can they be produced? Produced is discovery, testing, and manufacturing. The whole thing.
Sure. Well, yeah,
Amy,
sure.
We'll discuss, okay?
Yeah, we'll discuss. Yeah, whatever.
Why are people who have had both vaccines getting infected?
Because the vaccine only prevents severe disease. It doesn't prevent infection.
Yeah, vaccines in
it's
general
not really, I was going to say, it's not a concern.
In fact,
but
yesterday,
leave it to other people.
Yesterday, Fouché said the flu vaccines do not prevent infection. You get infected all the time. You just don't get disease.
Well, yeah, for sure. But you don't walk around getting PCR tested either.
No, that's a great point. If you did, you would see that you got flu in the winter.
Right. So, we should stop the testing in some respects. I know Daniel says never miss an opportunity to test, but it depends on who you're testing.
Oh, thank you, JCU 2803, for your contribution to the incubator, the BSL-5 incubator. Can you tell me why you don't think any variant will ever evade the vaccine so that serious COVID is back on the table?
Because the immune responses in the people who've been vaccinated have been demonstrated to evolve with the variants.
Yeah, the T cells are still effective against the variants.
Yeah, but even B cells have been shown. I believe that is Theodora's work, has been shown to evolve against the variants. So that even if you get vaccinated and you get infected with a variant, it is the variant protein that is being presented to stimulate the immune response, right? You're going to make antibodies directed against that because the vaccine antibodies are already, the vaccine stimulus is gone for months.
And Ron Fouché said something interesting yesterday. He said, okay, so you were vaccinated with mRNA vaccine and the ancestral strain you neutralize at this level, and then the Delta variant is down here. He said, but who knows? Maybe that's still, maybe for protection, you only need to be down here because the assays are not standardized. So he said people are worrying about the decrease in neutralization and they don't even have it calibrated.
You want calibration? That would imply organization of a field and not a free-for-all. Welcome to the world I live in.
All right, my nephews and other kids 1+ years old, all down with RSV, respiratory syncytial virus. Why in the summer? Why now? Spreading like crazy. Amy, why is this spreading now in the summer?
Because we came out of lockdown in the summer, and these children have no immunity. And so, yeah, it's spreading all over, and then it'll eventually go back to the winter. But yeah, Amy, it's out of control.
By the way, Amy and Susan, we have talked about RSV in a recent TWIV. It's an RNA virus, enveloped RNA virus with a negative strand genome. And it targets young kids. I care. It targets young kids.
Yeah, but it targets old people.
Oh, it preferentially
it targets
infects
old
young
people
kids.
Too. No,
it doesn't target them. No.
Yes, when you had Jason McClellan on TWIV before I lit into him.
But you didn't like Jason. You didn't like him.
No, I don't. I don't like him at all.
Okay.
I don't. Yeah, but he talked about it, and it targets old people. also.
By the way, here's an interesting finding. Fouché published a paper where they went into a hospital with a lot of kids with RSV infection, and they put an air sampler by all of them. Very little virus in the droplets. So they think it's mostly contact transmission among kids. Thank you, John, for your contribution. That's what I told you they explained, but we didn't have to, apparently. Do we have any new information on long haulers? Medicines to avert blood-brain barrier? No, we don't yet. But
why do I need to avert the blood-brain barrier?
Blood-brain barrier tears from the spike. I don't think that makes tears in the blood-brain barrier, does it, Amy?
No. I don't know where that came from.
See,
probably because people, once again, because science isn't done right and some idiot published a paper with brain organoids.
What's done? Yeah, so JEJV likes what it's done to science. Damn right. But Tom says, not so sure about that. The larger issue with science is cultural decay. It's been growing for decades. Do you agree with that, Amy? There's been cultural decay in science?
What is cultural decay?
The decay of the field, essentially. The decay of the way the field is progressed in the past.
No.
No?
No.
Amy, does COVID-19, SARS-CoV-2 cross the blood-brain barrier?
No. It's work by Madeline Lancaster. It hits, I think she thinks it hits some of the epithelial cells at the choroid plexus. Does not cross the blood-brain barrier.
All right, I don't know about this number. mRNA vaccine efficacy is only 62% against mild and asymptomatic. So we don't really care about asymptomatic. Because as we said earlier, the vaccines are not meant to prevent infection, which is what an asymptomatic infection would be.
So is a mild. I mean, who knows what a mild and stuff is?
Yeah. I mean, mild to Amy is probably killer to me, right?
Probably.
Do you have a high barrier of pain?
I broke my foot, Vincent, and I didn't take any time off. What do you think?
I'm not going to say what I think.
Was I not here the following morning?
You worked for 2 years with pneumonia, as far as I remember.
I worked 5 years with pneumonia. What are you talking about? I was sick 160 to 200 days a year in Montreal. That's not happening.
Amy needs hotkeys to splash "you think" and her other phrases across the screen. Yeah, I agree. I agree.
Okay,
we'll have to set her up with it.
Okay.
Amy, is it wise to vaccinate children?
Yes.
Absolutely. I mean, they can get multi-inflammatory syndrome. You don't want that. Multi-system inflammatory disease, whatever it is.
Look at all the stories that Daniel tells about, like, some kid got infected, didn't really develop disease, 3 months later can't play soccer anymore.
Coming back to my lament about the world being vaccinated, Earl of Mannheim says, isn't that what COVAX is for? Yes, I hope it works. I just don't have a lot of faith. On the other hand, Amy Ann says 2022 is too late to vaccinate. You said most of the world would be vaccinated.
Well, since we're halfway through 2021, 2022 is less than, half of 360. So it's less than 190 days away. So doesn't seem to me like that's a good it doesn't seem to me like that's an argument.
My neighbor said to me today, if you get your children vaccinated, you will have blood on your hands. I was so angry. I don't blame you. That's terrible. People that say
I
things
don't
like that.
understand that at all. I don't get that one.
Do you have any ideas what might happen in the UK with the release of nearly all restrictions? Well,
they'll have a surge to some degree, and then it will go down.
Yeah, the unvaccinated will be infected because there are a lot of them. So, yeah, there'll be a surge. And they'll blame it on some variant. You'll see.
Maybe it could be variant mu.
I have a great deal of confusion surrounding Marek's pathway. I don't know what you mean by Marek's pathway. I know Marek's disease herpes virus of chickens, but I don't know another Marek. Do you, Amy?
No, I'm not sure what he's talking about.
I thank you for alleviating my fears on the variants. Well, that's good. I'm glad that it helps, because that's what we're trying to do. Is the press is kind of out of control with the scariants.
Yeah, well, the press gets fed from people who don't know anything, like getting virology advice from an anatomical pathologist or a what was the person?
Neurobiologist and an
Neurobiologist.
anesthesiologist. Yeah,
Oh, yeah.
Jeffrey was arguing with an anesthesiologist on Twitter who said, of course the variants are more transmissible. They're spreading. Okay, Amy, 2% of cases result in long COVID. Does that number seem right?
I think so.
So in the U.S., we have 12,000 cases a day now. And a lot of long COVID. I think Daniel referenced an article last week. It's our next national health disaster, for sure. That's why no one should get infected.
Yeah, but people should actually put money into researching it, but it's going to be hard to do because it's going to be very hard to model, because of all of our models are really of absolute severe disease.
Yeah.
We don't really do models of wishy-washiness.
That's a great saying. We don't do models of wishy-washiness. That's great.
We don't.
I like that. That's another T-shirt. Amy is full of T-shirts. All right, this is a good one. Could we discover a virus that is completely novel? I mean, not related to any virus we know. Could we do that, Amy?
Sure, we can isolate a novel virus not related to anything we know.
So, Amy, hear me out on this. So when they do metagenomic sequencing, 90% or so sometimes is dark matter. They can't identify it because it doesn't have any homology with databases. So it could be that some of those are novel viruses, right?
For sure.
But here's the question,
But you have
Amy.
to be really careful.
Yes, because?
There's a lot of sequencing errors.
Yes, absolutely.
And you can't. So a certain individual who published a paper and another person who commented and sent emails before thinking things through violated the first, and another person who published another paper about RNA and DNA all violated basically the two cardinal rules of bioinformatics, which is crap in leads to crap out. And you are only as good as the database that you search. And no database is complete. So for instance, if I were to do metagenomics on a bacterial microbiome sample and the person had been diagnosed with chlamydia, I would never find it. Because the primers that we use are not homologous enough to pick up chlamydia. So you would come and say to me, my patient has chlamydia. And I would come and say, no, your patient doesn't have chlamydia. And then you would say to me, well, let's go to the microscope and look at the bug. And I would say, well, your microscope is broken.
Hey, Amy, here's another idea. Amy, here's a T-shirt. Amy is full of T-shirts.
It's funny.
Next time I'm arguing with you, I'm going to say you're full of T-shirts. That's great.
Yeah, I am. I need more T-shirts.
I've noticed a big drop-off in people wearing masks over the past 2 weeks. Yep, I have noticed that. I'm often the only person around. You noticed that, Amy?
Depends on where I am. So this morning when I went to go running and I got on the elevator to go down on 168th, every person in the elevator was wearing a mask. But when I got on the subway to come up to take a shower before I came to the lab, 2 people in my car were not wearing masks. So it depends.
Not good. in TWIV 659, Drosten says 34 degrees is better temperature for growth of SARS-CoV-2. Is that correct, Amy?
He might have said that. I don't remember, but that is probably true. So when people talk about respiratory viruses being temperature sensitive, they kind of ignore the fact that as you inhale air, you warm it up. So what goes in your nose at like 30 is definitely not 30 when it goes down into your lungs and stuff. So even though your chest gets colder when you have a respiratory infection, it's really pretty close to being like deep into your chest. The deep down lungs are pretty close to 37. Yep. And the airway is close to 34, which makes it really problematic when people talk about my virus and they say, oh, it doesn't grow at 37. And it's because you have some variant or some isolate that grows at 37 that it now has the property to neuroinvade. You know the crap I'm talking about?
Mm-hmm. I do.
And I just want to say, maybe you guys should take a physiology class.
Thank you, H. Zoo, for your donation. Yeah, we'll have an open house for sure. Yep, we're going to have our fans come and schmooze with them. By the way, your mug is in the mail. And those of you who have contributed on Patreon, I'm really slow at getting your gifts out. So just if you send me your address, I'll get them out. Because H. Zoo asked for hers, and I sent it out. In a moment of trying to be efficient.
What, did you have a stroke?
I am a stroke, yes. At least I can laugh about it. Not the stroke, getting things done inefficiently. Amy, I'm a couple of miles from vaccine complex in Canada. It started out over a century ago as a farm with horses used for antitoxin. Problem was mRNA makers wouldn't come to Canada. Now they will.
Okay, well, so you could be right, but all of the articles don't support this. And the people that I've talked to in Canada, and I understand that Quebec is a province and a country on its own. So I've been yelled at enough by saying this is what happens in Canada because my friends are in Quebec and they don't like that. But from a clear article in the Canadian Daily Mail or whatever the newspaper was, it said that there was no vaccine manufacturing plant and it wasn't going to be up and running until 2026. Remember that, Vincent?
Yeah, I do. But here's another person, ALC73 Canada, is supposed to be getting a Novovax facility before the end of the year. So,
well, they have less than 180 days. So let me know how that works out. Because I don't know anybody who can build a factory in that fast.
Probably
getting
in China,
all the permits.
They could do it in China.
But this is in China.
Yeah, I understand.
China, you don't need the bureaucratic permits and crap. And we used to laugh in Montreal about like how much of all the permits, because it was like you needed a permit to pour cement and then you needed a permit to dig up the cement.
So, Amy, if vaccinated individuals are still at risk for, I would say, getting infected, why are some businesses only mandating that the unvaccinated wear masks? It's a good question.
Because the unvaccinated has a much higher chance of getting severe disease. Vaccinated individuals are going to be protected against severe disease. So getting infected with SARS-CoV-2 is inconsequential. It's whether or not you're going to develop severe disease and cause an overrunning of the healthcare institute. I think people need to recognize what the point of the masks were and physical distancing. It wasn't to prevent you from getting disease. It was to prevent a big surge in hospital cases where everybody couldn't be taken care of. Had nothing to do with disease.
Preprint news cycle has been misused. That's another problem with science, right? Bloom deleted sequence paper. Peer review is essential, not propaganda by mainstream media, like how lobotomy got mainstreamed and won a Nobel Prize. What do you think about, Amy, have preprints been abused during this pandemic?
abused in the context of what they've been put out there, and then they've been taken as gospel.
Yes.
Yes.
Especially by news media, right?
Well, it's not just news media, right?
It's other scientists.
Yeah.
Can you rate on a scale of 1 to 10 about the dangers of nanoparticles?
Nanoparticles of what?
Well, so here,
like dust is a nanoparticle.
Yeah, well,
it's like a 0.1.
He's talking about lipid nanoparticles because he's talking about the aluminum used in vaccines, which is in some vaccines as an adjuvant, but it's not dangerous. The safety assessments have been done. There's not a problem with the alum in some vaccine adjuvants. But the lipid nanoparticles that constitute the mRNA vaccines are fine. They've been tested. I like this one's name. Je suis fromage. I am cheese.
It's good.
Have you considered speaking with pharmaceutical reps about receiving a commission on vaccine sales? No, I don't want to do that. I don't want to have contributions from companies that sell vaccines or drugs. Then people will say I have a conflict of interest, right? Which might be a reasonable argument, even though I don't. I've been listening to Twiv from 2012. Question about vaccine-induced polio. Is it that people who have vaccine do not get polio but can pass on the reverted virus to unvaccinated? Mm-hmm.
Yeah. That's one of them. But there are people who get vaccinated and get VAP. And that's not because it was passed on to other people. That's because it reverted and they have some host variant that makes them susceptible.
Yeah, so the VAP is in the recipient, as Amy said, and also in contacts. You get it in both. Tom, loving the Twivs, 95% ethanol. I was wondering if anyone would get that, Tom. You know, it's kind of a inside baseball thing, right? Amy gets it, right?
I do.
A lot of people know that stuff, and I thought it would be funny, right?
It is good.
Why does COVID hit the elderly with senescent immune systems harder than middle-aged people when it seems to be a disease of immune over-response to infection?
Well, you didn't have a response to begin with to prevent it. And you're not totally senescent. You're just dampened. And so you don't mount a full response, and then you have other complications due to that.
Peter Hotez's BioE Corbavax subunit vaccine is RBD only. Why do they expect this will work against variants? Amy? Amy's a fan, by the way.
For sure. So, as I said, you get vaccinated, you make an immune response. Then you get infected, and the variants get digested. They get presented, and you have a response. Then it gets enhanced by presenting to enhanced by presenting what the variants present. So now you've specified it for the variants.
Yeah, I think it's probably not a good... Pfizer tried an mRNA RBD only vaccine against the full spike, and it didn't work as well in their early assessments. I think there are much fewer epitopes, right? So it's not good.
Well, right, but that's not what the question is. The question is, why do they expect it to work again? Why do they expect an RBD variant vaccine to work? And the reason why is because you have an enhanced basal response, then you get infected with the variant gets presented, and now you have a head start to fighting it off, and then the immune response then becomes catered, the next round becomes catered to the variant. Has nothing to do with the number of epitopes or whatever.
I'm just saying I don't like it because it doesn't have enough epitopes, neither T or B cell epitopes.
Yes, but it doesn't. That's not the question of whether or not
I don't care.
Like it.
As you know by now, after how many years, 20 years, that I don't always look at the question.
Well, then there's no point in taking questions. Like,
No,
that's just ridiculous. Okay,
I let
that's
you
just
look at it.
ridiculous.
They're saying that nanoparticles go in the brain. Now, so I don't know who they is, but it's not true. They don't go in the brain. They've been completely properly evaluated, first in preclinical studies in animals in the lab, where they put lots and lots into animals and see where it goes. And then in people, millions of people have gotten these. It's not an issue. We would have seen it.
And if they're worried that nanoparticles get into the brain, I wonder what they're worried about when alcohol gets into the brain.
Oh. Does alcohol go in the brain, Amy?
Why? Yeah, I mean,
Yeah. Wouldn't part of viral fitness involve being less lethal to hosts so it can proliferate more? I think yes, that's possible. That's a very good idea. And we think about that all the time. Yeah. Reduced lethality. Reduced lethality is a good way to be. I got you, Amy. I got you. One more question. Let's find a good one here. They're all good, but I'm finding one.
I need to go home and work on the grant before I fall asleep.
Oh, you want to answer this one? Why is vaccination the only solution?
Because humanity is stupid.
What does that mean, Amy?
You can't get the horse back in the barn, right? So like you could have done surveying, you could have done antivirals, you could have had an intervention policy that was more effective. All of these things would have made you less dependent upon a vaccine, right?
Mm-hmm.
But we didn't do any of them. Consequently, humanity is stupid.
And that you want to leave on that note?
You think that that's bad? All right, I'll tell you. We could do one more.
It's a bit harsh. It's a bit harsh.
But it's true.
It's true. So we haven't pursued other approaches. That's the problem, Jen. And we're way behind in antivirals. So we have to do vaccines. And the problem is, Jen, maybe you don't like vaccines, but there are a lot of people who don't like antivirals, so you can't please everyone. Okay, here's the last one. What do you think about Israel reporting Pfizer is less effective against Delta? Infection. At least you put that in there, Martha. Very good.
I don't think that that's correct.
You don't believe the data?
I haven't seen the data. Have you seen the data?
No, but as we said earlier, who cares if you're infected, right?
Right.
I bet in these people they didn't get moderate to severe disease, and that's all that matters. So
plus, so I'm not really that concerned.
Yeah, neither am I. All right, Amy, thank you very much as usual.
Thank you very much. All right, tomorrow, grant day.
And we have to change the CO2, right?
Yeah, change the CO2 budgeting.
Okay.
Grants.
Thank you again.
All right.
Good night. Bye.
Good night.
Oh, how come we have this thing here? Okay. So the problem with the Israel report is that they're looking at infection, and most vaccines don't prevent infection. They prevent disease. So who cares if the early after vaccination, here's the thing, early after vaccination, your antibody levels are high, so you're going to prevent infection. Then that wanes pretty quickly, and that's what they're seeing here. It doesn't matter. And this is the nuance that they don't get, so the press just runs with it, and people get scared. Oh my gosh, if I get vaccinated, it's not going to be of any use. It's not true. The infection doesn't matter. Tell me how many of those people died. Probably none. And that's what we care about, serious disease. What's the verdict on Sweden's strategy? Actually, I don't have a verdict. I'm not sure what they did, because every time we talk about it on Twiv, we get scolded that we don't have the right information. So I would like to get someone on to talk about it who knows what's going on. And, there's so many countries with different strategies that it could take a while to do that. Calgary Stampede. Oh, I've heard about that. My wife's family is from that area. Calgary, Edmonton, Grand Prairie. To enter, you have to prove vaccination or take a quick free antigen test. That's interesting. All right, that works. Of course, you can fake vaccination proof, right? So here's my vaccine card, right? Look at this. It's not in focus. COVID vaccine record card. There we go. I mean, this has got CDC imprimatur on it, but who knows? You could fake this, right? And then there's something on the back. All of this is fakeable. And there's just handwriting about when I got it, right? I think proof of vaccination, I'm not sure how good that is. Thank you, Susan, for your contribution. Really appreciate it. Are vaccine companies making quadrivalent vaccines? Well, they're making individual mRNAs for the different spikes and testing them and seeing their immune responses and so forth. Frankly, I don't think we need them because we have no idea what level of neutralization with antibody, vaccine-induced antibody you need in order to protect. So if it goes down with a variant, how do we know that matters at all? It may be abnormally high to begin with. As I said, the T-cell epitopes do not change in the variants. So that tells me you don't have to change the vaccine. Now, I've heard vaccine manufacturers talking about making second-generation vaccines that are a little broader. So we just use spike for making the process go quickly, but maybe we should include other proteins to get more T-cell epitopes. I think that's not a bad idea. But this idea that we're going to have a booster is absurd. It's not a booster. It's a vaccine. Like a flu vaccine is new every year or so, right? It's a new vaccine. We don't call them boosters. Why are we calling them boosters? I don't get it. But as, you know, Amy said, there's a lot of weird stuff going on. Will the mRNA vaccine cause any damage to our immune system? No, it's like another foreign protein coming in. You make an immune response to it. Our body's immune system needs to be worked out, right? From day one when we're born. And it makes it better. And this is just another antigen. Well, I don't know why it would be any different. So there's no evidence that it's an issue. I think that's the most important thing. How do you produce small quantities of monoclonals? All right, so there are at least two ways. And in writing this grant, Amy has explored these. So one thing you can do that's pretty cool is you can immunize mice, say, with your virus, and then take blood and isolate the B cells. And then you can use a peptide that is conjugated to an oligonucleotide to pull out those B cells and then sequence the antibody locus. And then you can take the genes encoding antibodies and express them in another cell and make the antibody. That's the modern approach. The old approach is you would immunize mice, you would remove their spleens and take out the B cells, fuse them with a immortalizing cell line, and then get hybridomas that make antibodies. So that's how you produce small quantities for research in the laboratory. You follow hundreds of channels, and you will be pleased to know that yours is most viewed. Thank you. I appreciate it. I mean, I'm not a huge YouTuber, right? I don't have millions and millions of views, but I think the people who watch appreciate it. Hey, we just hit 89,000 subs. Thank you, everybody. Maybe we could hit 100 by the end of the year. It's just a nice number, right? Is there a difference in post-translational modifications between Novovax and mRNA vaccines? Yes, most certainly. The Novovax is produced in insect cells, and so the mRNA vaccine is injected into you, and your cells are producing the protein. So it's very likely that there are different post-translational modifications. The real question, as you asked, does it make a difference? We don't know. People may be looking at that, but so far I haven't seen any data. How long do you think it will take to find the spillover? Dr. Livingston, yeah. So what you mean, Jefferson space, is how long will it take for us to find the immediate, or as they call it, the proximal origin of SARS-CoV-2? which means the virus just before it jumped from bats into people. For SARS-CoV-1, well, they didn't actually find that, but they found in a cave of bats in China viruses with all the pieces that could make up SARS-CoV-1, and that was good enough. So that took almost 10 years. So it could take at least 10 years. But the other problem is these Rhinolophus bats that harbor these beta coronaviruses, they have a huge geographic range. So looking throughout it, at least 5,000 square kilometers, it's going to be quite a chore, right? Thank you, Daleen. Appreciate it. Really like that. I'm just a scientist and an educator doing my thing. Hey, no BS level 5. That's great. I like that. The depressing thing is everyone is talking about Delta, but Lambda is the next most serious. It's not going to be the last. I don't know why you say it's serious. It's just another SARS-CoV-2. I mean, they will keep changing, as long as we let the virus circulate. But look, influenza circulates because not everyone is immunized. We get variants all the time. It's going to be the same thing. It's not depressing. We can handle it. What's your prediction for COVID? I'm not sure how that's going to be handled, right? Isn't it? They're only letting people from Japan go to the Olympics. They have low vaccination rates. If they don't require vaccination, it's going to be a problem. Yes, for sure. Important point. Most media seem to miss that. Meds do not prevent infections, but help lessen disease. Yes. Polio vaccine doesn't prevent you from getting infected, prevents paralysis. That's how they're tested. So the Israel report that says prevention of infection is decreasing is a bit absurd because the vaccines were never tested to prevent infection. They were tested to prevent COVID of various severities. No, the media has, well, I don't want to blame them because Amy would say it's also the scientists' fault for not explaining it, and that's true. The incubator. What is the transmission if you get infected with COVID after a Pfizer vaccine? We don't know. It's probably decreased, I would guess. So I've been saying tonight the vaccines don't prevent infection, but I do think they reduce virus replication sufficiently to prevent transmission. And those studies, a few studies have looked at that, and there seems to be an inhibition of transmission, but I don't know the final numbers, Christina. We'll find out for sure. Are there papers to show the antibodies from jabs are protecting against new variants? Yeah, there are a whole bunch. They're mostly preprints. There's some, I think they're all preprints. Maybe a few have been published. But yes, absolutely, people are looking at neutralization assays for that. You bet. You bet. So here we go, Martha. We just talked about that. I don't think it's less effective at preventing infection, but not disease. A little nuance that's really important. What influence does change in a few of the epitopes of an antigen have on kinetics of immune response? Well, the protein is largely the same, so you will have a memory response to the original protein and then an altered response to the individual epitope. So I don't think there's any difference. If it's a memory response, the kinetics are more accelerated, of course. In the future, can you talk about viruses in the gut microbiome? Yeah, whenever I'm going through papers for TWIVs, I always look at those. I'm waiting for a really good one, but we will. So what is today? Wednesday. Tomorrow, Thursday, I have to pick the papers for Friday's TWIV. So I do that pretty much. I don't mind doing it. I often consult Amy because she's right down the hall. Did you find any good papers lately? And then she may have some or not, in which case I'll go back and look at them. What types of serological assays would determine vaccine effectiveness for immunocompromised? It depends on what kind of immunocompromise they have, right? So there's some where you don't make B cells. There's some where you don't make T cells. And there's some where you don't make both. So if you can't make antibodies, you're not going to check for them, but you could check for T cells, although unfortunately it's harder to do a T cell assay. And if you're T cell compromised, you could look at antibodies, for example. Ralph, thank you for your contribution. Your BSL-5 incubator should be for gain of knowledge research against disinformation. What a great thing to say. I love that. Let me give it a star so I can remember. So first of all, I love how memes arise during our little chats, right? One day it was Right Amy and I don't know, whatever, but tonight it's BSL-5 incubator. When you get 500 people together, good things happen, right? A lot of creative juices flow. I always think that's why I like teaching, when you get students together and you have a good conversation. Gain of knowledge research against disinfo. I love it. Gain of knowledge research. I have to write it down because I'm not going to remember to look at this. Gain of knowledge research against disinfo. It might be a T-shirt, right? Might be a T-shirt. Thank you, Steph, for your contribution. Of course, all your contributions go to the incubator. Thank you, Crediolopathic Remedy. A sign displaying BSL-5. So we have an incubator sign. Excuse me, we have an incubator sign, which is really cool. I mean, if you were going to make an incubator sign, what would it look like? Let me see if I can quickly find it here, because I did make one. Too many folders on my damn computer. I don't know. Let me search for incubator. Incubator in my hard drive. Here it is. I found it. Holy crap. Holy crap. Look at that. I found it. So I can show it to you guys. Oh, you're going to love this. Look at this. Let me close this and go to the thing. You like that sign? It's like, the LED on an incubator. So I want to make that and put it on the door. And then we'll have, the microbe TV and Parasites Without Borders. Let's discuss for the T-shirt shirtless. Yeah. All right. So now I understand when Amy says let's discuss, she doesn't like it. Okay. I had both vaccines. I'm fearful to travel. Well, what vaccines you had? J&J and either Pfizer, Moderna, right? You had 2 doses. Yeah, you can go on the plane, wear a mask. Don't worry about it. From Florida to upstate New York. Yes, wear a mask. You're vaccinated, you'll be fine. Is there anyone working on architectural designs that are beneficial to mitigate spread of viruses? Absolutely. HVAC tech, putting in far UVC lights, encouraging sunlight percolation, which has good UV, and through windows that allow UV to pass. A lot of things going on that I'm not aware of. I don't follow that space. Dixon should, but he doesn't follow it either. Can you explain why the new RSV vaccine works and doesn't cause enhanced disease like the earlier one did in the 60s? So what happened with that one, the 1960s RSV vaccine was treated with formalin, and it destroyed a key protein on the particle, and so the kids didn't make antibodies against it. And so there were a number of different issues, one leading to excess inflammation, one leading to infection. So that's the problem. And the new vaccine is an mRNA-based vaccine, as Amy said last time, and that's not going to have a problem with that. So that's why it's better. But I tell you, that vaccine in the 60s delayed work on an RSV vaccine for many years because people were afraid to touch it, right? It was really a bomb. Thank you, Tom, for your contribution. Oh, it was the country, not the field. Sorry. Cultural decay is another way of saying regulatory capture, which is undeniable. So I think in science there's cultural decay also. Maybe it's not decay. Maybe decay is not the word. I mean, all right, let's just say it's change, because I trained in the 70s and 80s, and things were very different than they are now. You know, you published a paper with 1 or 2 people on it. Now you publish a paper with 100 people. And I don't know how you collaborate with so many people and figure out who's first and who's last. I don't know how you could do that. But also, publication is very, very difficult. The journals have made it hard to publish in certain journals. They want to restrict. I'm on a journal board, and we got these emails from the editor saying, "Oh, our impact factor was really good last year." You know, the impact factor is how many times your papers are cited in other papers. So the higher, the better. But we can do better if we accept fewer papers. Can you imagine artificially raising the impact factor? It's just the point of science is to publish information that helps people, not to get your bloody journal a high impact factor. And this was a nonprofit journal. It doesn't even make any money. It's just for the egos. Unbelievable. Yeah, this is a report out of Israel, which is totally bogus and misleading. And, you know, if I were efficient, I could have sat down and made a 10-minute video in 2 hours that explained it, and that would help. That would really help. I can't do it on my own anymore. I don't have time. I'm teaching right now and all that. But that's what I want to do. I want to respond to stuff like that. And people will see it and maybe spread it and say, oh, I get it, because the press can't explain it to them. Now that we know there's aerosol transmission, there's a 6-foot rule. I think the aerosol, which means the tiny drops that go a long way, this is pretty rare. I don't buy that it's the norm. I think the norm is still about 6 feet. Yes, I think it's fine. Thank you for your contribution. Really appreciate it. I really appreciate it. The idea that some low GDP countries can afford the kind of tens of billions the West has spent is thinking sadly. Yeah, I agree. And I think we need to help them. I've always been of the mind that we should help others, right? And that's why I give away my knowledge, my course, my pods. The only thing I don't give away is the textbook, because I don't publish it. But if I published it myself, I'd give it away. I want people to have knowledge, and they should have vaccines as well. You would like more antivirals? We could have, before this pandemic began, we could have developed them and had them ready for sure. They're harder to do than vaccines. Marty, thank you for your contribution. We make you smile. Thank you. Thank you very much. Is it possible to make a vaccine in a pill? Yeah, for sure. So, you know, as you know, you can make a pill that will go through your stomach and not melt, right? It will melt in your intestines and release a virus, say, that's a vector that will infect your intestinal mucosa. You can do that with adenoviruses that infect the gut mucosa. And then, you know, you immunize one mucosal surface, you get the other ones. You have multiple mucosal surfaces, as you know. You have the gut, and you have the urogenital tract, you have your eyes, you have your respiratory tract, alimentary tract. So, yes, it can be done, and people are working on it. It's a very cool idea. I'm looking for questions with my name or Amy's in front. Yeah, Amy's full of T-shirts. I am not gonna forget that. Do I think viruses are the origin of life? Yeah, I think viruses started it all in the pre-cellular stage of Earth. I think, you know, as the Earth cooled and organic materials began to form by whatever reactions, small self-replicating molecules arose, and those eventually became RNAs that self-replicated, but there were no cells. And eventually they evolved. And then, so it was an RNA world only. They're not viruses because they're not requiring cells yet, because there are no cells. But eventually proteins evolve, and then among the proteins are enzymes that replicate the RNA. And then, whether you need cells or not for that, I don't know. That's a good question. Then the reverse transcriptase arises that copies RNA into DNA, and boom, now you can have DNA-based cells. Why? Because RNA is too small to make a cell. Cells are complicated, and RNA can't get big enough. The biggest RNA that we know of is 40,000 bases. You know, chromosomes are millions of bases. So I think in that sense, yeah, viruses predated everything. We are all viral. You could look at it that way. You could look at it that way. Yeah, I got it. I just answered that. I try and go methodically through this. I know it's inefficient, and some of you get mad and you give me a thumbs down. I'm very sorry. We have a lot of questions, which is great. I try to get through them all. When you say 2% result in long COVID, it's including asymptomatic. However, with the caveat that we're not so good at diagnosing asymptomatic, right? Because we don't test everyone. So it may be an underestimated fact. Is Delta more dangerous in terms of mortality? I haven't seen any data that would suggest that. Now, Tony Fauci said it may be more deadly based on hospitalizations, but that's not a fair comparison. You can't say, oh, today there's more Delta hospitalizations, and 3 months ago when it was a different variant, we had fewer. You can't do that comparison. I think that's totally unfair. And that was done in the UK in the Alpha variant emergence, and it was wrong. It was totally wrong. So you have to be very careful of these proclamations of more this and more that. In science, things take a long time. And unfortunately, in a pandemic, as we pointed out in our op-ed, answers take a long time to be figured out. Yeah. Someone said, how do I get the op-ed? Send me an email, vincent@microbe.tv. I've been sending it out to people who want it. And yeah, if I haven't answered, just re-ask. Don't get mad, just re-ask. Isn't long COVID just post-viral fatigue syndrome? No, it's not. It's quite different. There are different properties, and people are beginning to realize that. I mean, it's an interesting corollary or parallel. That you get a viral infection, and then you have post-viral fatigue, MECFS, long COVID, but the features are quite different of the two. I had a stroke of efficiency. So if someone says, I need my mug, I will send it right then and there if I have 5 minutes. But as for sitting down and going through the 400 people that need mugs, man, that would take me hours. So I can't do that, but I send them out here and there. What made me fall in love with virology? There's a book called For Fever by John Fuller. I had graduated from Cornell not knowing what to do. I graduated in 1974. I got a job as a lab technician. I really liked it. I was doing a microbiology lab, and I read Fever. I said, Fever is about the emergence of Lassa virus, right? And I said, I have to become a virologist. And I have to say, I had gone to college with Ed Kilbourne, who was the son of Edwin Kilbourne, who was the chair of microbiology at Mount Sinai in New York. And he's the guy who engineered the swine flu vaccine in 1976 that was such a disaster. Anyway, I went to Ed's house, and Ed Sr. was there, and I asked him what to do, and he said, come get your PhD at Mount Sinai. There you go. And I loved it more and more as every day went by. Are antibody tests reliable? And when you get your IgG response to SARS-CoV-2, what do the numbers mean? It depends on the assay. They can be reliable. Depends on the test, right? Every test is slightly different, and they may measure them differently. But if you have your sheet and you want to send it to me, Vincent@microbe.tv, I'll take a look at it and explain it to you. I'd be happy to do that. My friend paid $1,000 something to rescue a dog from a China market. So there are pet markets in Wuhan because I know I read an article about them, and I'm not surprised. There's a picture in the journal article that we did on Twiv last week, and it has a stray dog walking around. So, yeah, it's not a gimmick. I mean, they have lots of dogs in China, and they shouldn't be running around, right? This is Ping from Pittsburgh. Could you explain the definition of viral fitness and the factors that affect it? So fitness, all right, survival of the fitness. Darwin thought that up. You have islands with a bunch of finches, and on one side of the island there are plants with hard shells, and a bird is born one day with a slightly thicker beak, and that bird is really good at eating those beaks. So that bird and all its offspring with big, thick beaks are more fit to eat those berries or seeds or whatever. So the same thing goes for viruses. Viruses can be more fit as well, which means they persist in the host. So they outcompete. So the bird with the harder beak outcompetes other birds for that food. So viruses that are more fit outcompete other viruses. Hence, alpha variant outcompetes the predecessor, and now delta's outcompeting everything else. It's all about fitness, not transmission. Transmission is different. Transmission is moving from one person to another. The problem is people see a variant dominating and they assume, oh, it's spreading, it must be more transmissible. No. If you want to show it's more transmissible, you better do the experiment, and it's not been done. So fitness, what is fitness involved now for a virus? So many things, but one of them, and I think this is a main one, is evading the immune response. In an immune population, if you have a spike that can evade antibodies, you're going to be more fit. You're going to outcompete other viruses. And that's what happens. And notice, the variants didn't start dominating until towards the end of 2020. They were all there in the population. You can find them in the sequences, but they were minute fractions of all the variants out there. Only when the world's population gained higher levels of immunity did the selection force exist to select for variants that could escape antibody, and that made them more fit. So that's one example of a fitness advantage. And that happens with influenza virus. Every year or so, new variants emerge that evade antibody responses. Only about 10% or so, that's all it takes. And those outcompete previous influenza variants. And guess what? Nobody calls them more transmissible because they're not. They're more fit. They are more able to dominate the population because they evade an antibody. And there can be other things. They can be less virulent, right, as someone suggested, and spread easier in the population because they allow you to be asymptomatic, perhaps, or mildly infected. And many other properties like reproduction, maybe levels, stability. I think the antigenic escape, though, is a big one. Okay, I hope that answered it. Have we all forgotten or changed our positions that masks protect not the wearer but everyone else? Well, they do provide some protection to the wearer, not as much as to the other people. So if you're not, non-vaccinated wearing a mask is not backwards because it will prevent you from being infected. I mean, there is some protection there. It's not just shedding to someone else. I think that's known. Thank you, Sandy, for your contribution. What's your best guess at how long the vaccine protection will last? So your last dose was August. Should it start to worry? No. You got 2 doses. You're protected against moderate to severe disease probably for years. You're not going to be protected against infection, but as we've said, that doesn't matter. So years. You have years. You can go see your grandbabies for sure. Absolutely. Some questions repeated, of course, but I did answer some of them, but that's okay. I had a reaction to Shingrix, the shingles vaccine. My arm got swollen. You'll have less protection. You should go get the second shot. You'll get swollen again, but I think it's a good price to pay. I don't know anything about how they vaccinate people in Iceland, but you could tell me. Let's go down and find some. Now it's 5 minutes before the end. Here we go. Ian Fraser. Send me an email, Vincent@microbe.tv. I will send you the op-ed. I have a copy of it. Thank you for your contribution. Really appreciate it. Pfizer's 93% effective against severe disease. That's all we care about. All they're reporting is 60% efficacy or efficiency against infection. We don't care about infection, folks. We don't care about infection. We want to prevent disease. Let's see some more super chats. We have to thank people. DR, thank you very much. What attributes of a virus make vaccine development more difficult? Well, in the old days, if you couldn't grow a virus in cells, you couldn't make a vaccine. But obviously now that doesn't matter anymore because we can make spikes, mRNAs. What attributes? So for whatever reason, HIV is particularly refractory to preventing by vaccination. And we're not really sure. And I think one of the attributes is that we don't know what protects. Is it antibody or is it cellular immunity or both, or is it innate immunity? I think that's a problem. I also think that viruses that have a viremia phase are easier to vaccinate against, and these respiratory viruses are tougher. But, I think we're going to be spoiled by SARS-CoV-2, just as we were spoiled by measles, because the vaccine is great, and the flu vaccine is not. And it's not clear at all why. Thank you, Susan, for your contribution. Really appreciate it. Sandy, I got your question. I'm zipping down here. Podcast about viruses, the kind that make you evolve. Thank you, Javier. I love it. Yes, you should read Fever. It's a great book, and it was out of print for a while, but now it's back on Amazon. You can get it in Kindle. I highly recommend it. The TWIV Book Club, sure. Can I be part of it? I can just listen. Thank you, Overjoyed, for your contribution. Appreciate it. I'm sorry that you've had so many troubles with COVID. I hope it all works out in the long run. Thanks, Joe, for your contribution. Really appreciate it. Vincent, the yearly paper said 80% of forward transmissions are from 15% of cases. That's right. Even if a variant was 50% more transmissible, wouldn't the chance variance in that 15% always dominate? You would think. And the other thing is that I lost my train of thought. Anyway, you're right. Yes, I agree with you. Yes, the other observation is that variants travel around the world, but they don't always dominate where they go. So if they were, that to me is a sign of fitness competition. If they were really completely more transmissible, they would dominate everywhere, but they don't. Yeah, that's right. People tried COVID-1 vaccines, but they didn't get very far because nobody was interested. The disease was gone. It was over. So why would they want to invest? But now we have nonprofits like CEPI and Ready that are presumably going to bring vaccines and antivirals to phase 1 that no company will touch, and maybe they'll be ready. So maybe the next pandemic, in fact, will be prevented. Ron Fouché thought it would. We need liquor sticks for all. That would be great. Liquor sticks for many infections would be great. All right, folks. That's it for another Wednesday night live stream. I hope you enjoyed it. I hope to see you back next week. For your listening pleasure, a little Nadia on the piano covering some Fiona Apple song. Please, everybody, stay safe and see you next week.